thing. It's just one gene here, and it makes a protein that brings itself back to its own gene and causes that gene to be copied at new locations, or at least snipped out and appear at new locations in the genome. And where the new copy of the transposon gets inserted in the genome is completely random. It could pop into a bit of non-coding DNA, or it could pop into a functional gene in the DNA. Retrotransposons are kind of similar in sequence, but or I should say in concept, but they kind of have a twist to them. There's an RNA intermediate involved in the process with the retrotransposons. And there's also the reverse transcriptase enzyme. And remember that, we've discussed that a couple times. That's the enzyme that can take a piece of RNA and convert it take a piece of RNA, convert it into DNA. So let me draw one of these retrotransposons for you. There's the nucleus again. There's the rest of the cell. All right, so here is a chromosome again, and here is the retrotransposon, like that. So the retrotransposon also has a promoter, so it makes an mRNA. And the mRNA eventually exits the nucleus. And then, well, you know what happens to mRNAs? They get made into proteins, and the mRNA encodes for a reverse transcriptase protein. And so recall that this enzyme, reverse transcriptase, can grab hold of mRNAs and make a DNA copy of them. But what's special about this reverse transcriptase is it's able to hone in only on its own mRNAs. Each mRNA from a particular gene has a particular sequence, and so there's some sequence within its own mRNA that it looks for. It ignores all the other mRNAs in the cell and just finds its own mRNA. But when it does that, when it finds its own mRNA, it does what reverse transcriptases do, which is make a DNA copy of it. So now you have a DNA copy of the mRNA. And then that DNA copy is what inserts itself into a new location in the genome. And if you think about it, it's the same sequence that you started with here. One strand was transcribed in mRNA, and then the complement of that became the cDNA. It eventually becomes double-stranded and plugs itself back in. And so if you think about what the end result is, it's a way of essentially reproducing it. It's just a reverse transcriptase gene that's reproducing itself inside the genome. It's kind of a circuitous path, but that's what it's doing. And since the mRNA is involved in the process, both encoding the protein and being the template for making the DNA copy of the retrotransposon, that's what makes it a retrotransposon, because reverse transcriptase is involved. Now, these ideas of these transposable elements jumping around in the genome seem so strange. People sought an explanation for this. Who remembers what type of organism we talked about before that carries reverse transcriptase enzyme as one of its genes? Viruses, yeah. There's certain types of RNA viruses, and remember, RNA viruses are actually one of the most common kind. Most of the viruses that affect human beings, for instance, are RNA viruses. And some of these RNA viruses carry a gene for reverse transcriptase. Viruses have other genes also. They have genes for the capsid proteins that make the shell around their chromosome. And there's other viral enzymes, genes for other viral enzymes that are on the viral chromosome. Let's see, oh yeah, some of them have this other proteins that form this little claw thing so they can attach themselves to cells. And remember this, when a virus infects a cell, the important part of the virus to get inside the cell is the chromosome, because that contains the genetic information for the cell reproducing the virus. And then once that chromosome gets inside there, it has very strong promoters, and so the cell starts expressing those genes, and some of the enzymes of the virus actually chop apart the host's DNA, so the host DNA doesn't compete with the virus, and then the cell turns itself into a virus-making factory and makes copies of the chromosome and makes new capsid proteins and whatnot until eventually the cell explodes and all the offspring viruses come out. Well, anyway, so remember that for some viruses, they carry the reverse transcriptase gene. And so what they think the origin of the retrotransposons is that some virus sometime in the distant evolutionary past infected a cell, and its reverse transcriptase gene somehow broke off from the rest of the viral chromosome. And the reverse transcriptase gene integrated itself into the host chromosome. And at that point, it couldn't escape anymore, because it no longer encodes capsid proteins or the other proteins that the virus needs to escape from the cell and infect other cells. And so it's almost like a mini virus, I guess I would say, that can still jump around in the genome. It can infect one chromosome to another chromosome, but it can never leave the nucleus because it just doesn't have the other proteins that the virus needs. So don't get me wrong here. Transposable elements are not viruses. To be a virus, you have to encode all the capsid proteins and infect from one cell to another. But they think they originated as part of a virus's gene, and so now it's almost as if they're a virus that's trapped in the nucleus that can infect one chromosome to another chromosome but never leave the nucleus. All right, so here's a thought. Imagine a retrotransposon where part of the gene was lost, and so instead of encoding the full reverse transcriptase, it encoded a non-functional version of it, maybe only half the reverse transcriptase, maybe only rev. And so given that it's making a non-functional reverse transcriptase, do you think it could jump around in the genome? Well, at first you might guess no, because you'd say, well, unless it was a functional copy of reverse transcriptase, it can never go back in and make a DNA copy of itself, and so that's not going to work. But there's a workaround to this problem. You do find some partial retrotransposons that encode only part of the reverse transcriptase and not the full thing, and these are called signs because they're smaller. Since they don't encode the full thing, they're short interspersed nuclear elements. Anyway, so they encoded a partial but a non-functional version of reverse transcriptase. But as long as some other retrotransposon is in the nucleus, as long as there's some other retrotransposon in the nucleus making functional reverse transcriptase, then this sign, this partial one, can get its RNAs copied. Because remember, this reverse transcriptase looks for certain sequences in the mRNA to go back and make a DNA copy of it. And so even if you only have a partial mRNA, this other functional reverse transcriptase will recognize its sequences and make a cDNA in it. And so it kind of blows my mind. It's almost like a parasite on a parasite. Here's a functional retrotransposon, and this one's sort of parasitizing its reverse transcriptase to make copies of itself, even though by itself it doesn't make any functional reverse transcriptase enzyme. One of the most abundant of these signs is something called the ALU sequence. I'm not sure where the name comes from, but it's a very short sequence. It's the one that's bracketed by these red letters right here. I think it's around only 300 nucleotides long. Well, it's the ones bracketed by the red right there. And it's exactly that. It's only part of the reverse transcriptase sequence, but it's so good at getting itself copied by other reverse transcriptase enzymes that it now makes up about 10% of the human genome, which, if you think about it, is about half of your entire functional genome in terms of making genes. So almost half of your genome size in terms of actual protein-making genes is made up of this ALO sequence. It's like 500,000 copies throughout your entire genome.
For partial retrotransposons to be able to use reverse transcriptase inhibitor, retrotransposons, they have to be the same.
Yes, yeah. So they're definitely related, and they have the same sequence, and so the reverse transcriptase is able to find the sign mRNAs and make those into cDNAs. Yeah, so they're definitely very related to each other, but one's a fragment of the other, basically. So I guess on this diagram right here, I might say some of these are the sign things. They're like partial retrotransposons, fragments of retrotransposons. Yes.
How often is this happening in our stuff?
Yeah, it's a good question. I tried to find that information because to me that's an important question, and very few sources gave a number for it, but one source did. It said that when they look at human beings, when they analyze individual people's genomes, about 1 in every 200 people has one new ALU insertion. And so given that you have like 500,000 copies of ALU, and only 1 in 200 people have evidence that it's moved, it doesn't happen very often, but it does occasionally happen. The take-home message there. Okay, well, let me think. Let me give you the quiz here, and then I will, I guess, see you on Monday. And we will have our last 2 meetings of the semester.